Thank you all. Good morning. I'm so happy to see all of you here and so many of your families and support people. I would like to welcome you to our symposium this morning. And I am Sylvia Newell. I'm the director of Michigan Sea Grant. And Michigan Sea Grant is one of the 33 national sea grant programs. So the National Oceanic and Atmospheric Administration, NOAA, runs a sea grant program in every state with a coastline. And the idea is to promote healthy coastal ecosystems, resilient coastal communities, and sustainable fisheries. And so NOAA helps ensure that by putting a team in every state that has a coastline. And it turns out the Great Lakes have more linear coastline than the Atlantic coast, the Gulf Coast, or the Pacific coast. We have about 4,500 miles of linear coastline here. And Michigan itself hits 4 Great Lakes, right? So I like to think of us as sort of the linchpin Great Lakes states because we connect almost all of the Great Lakes. And so that means then that our communities here tend to be really tied into the health of our Great Lakes and also our inland lakes because we've got so many thousands of them. And so one of the problems that we have is that as our coastlines are changing, which they really are on the Great Lakes, water levels go up and down more than people on the coast are dealing with sea level rise. So as our communities are impacted by changing quality, as we're getting more harmful algal blooms, as we're being impacted by much bigger frequent storms that are flooding out coastal towns, as we're dealing with rising and falling water levels in the lakes and how they hurt marinas, water issues are a really big deal in Michigan. And one of our goals as Sea Grant is we fund research, education, and outreach because we are trying to figure out how to help communities deal with these problems. But also we're trying to train the next workforce of people who care about these systems and how to help communities come up with solutions to them. And so we want that workforce to come from people who live in Michigan who are impacted by this as part of their daily lives so that, when you go home and talk about it, your family cares about it because everyone cares or should care about having clean water to drink, being able to go and enjoy our natural water resources. Hopefully all of you like fish and eat it, although I know not everyone does or enjoy fishing. And, the mission of Sea Grant is to promote all of this while trying to make sure that our next workforce reflects the full diversity of the communities that live in Michigan and are impacted by these changes. And so we are committed to a diverse and inclusive engagement to facilitate research, education, and outreach partnerships. And what this means then is we're trying to take the people who live in communities and connect them to science, whether it's because you live in a community that's on a coast or whether it's because your business and your economy depends on the health of the lakes around us and within Michigan, I guess. And essentially we all benefit. For example, NOAA is actually a part of the Department of Commerce. I'm not sure if you knew that because most people think, why would it be, right? But it's because the shipping, especially through the Great Lakes that happens, is incredibly important to our economies, as are our fisheries. I mean, billions of people around the world eat fish, and so it's a multi-billion-dollar industry. So it's really important to the health and economy of the Great Lakes states. So this internship then is our way to connect undergrad students with private businesses, local governments, state organizations, and environmental nonprofits to try to help ease that transition from what sometimes feels like a kind of esoteric curriculum. As an undergrad, you're like, why do I care about this again, right? I don't want to get up at 8 a.m. to go to this class. But then hopefully by going to this internship, you understand how building the fundamental principles within your field will help you be prepared and connect you to a job later on. And by having these internships, we're giving you that experience to hopefully be able to get a job in one of these fields and help Michigan's coastal communities and coastal ecosystems. So we're trying to really teach you all to be stewards of the environment and help you in turn help your communities become stewards of the environment. Now for a sad moment, I know that all of you were initially coordinated for your internships from Dr. Mike Fraker, who was our research program manager, who unfortunately passed away in April. And just to talk about Mike's commitment to students, he literally got up out of the hospital and came to the admitted students luncheon about 2 weeks before he died, so that the students he recruited would know that he was that committed to, their success of their education. And so, yeah, we're sad. And so we are taking steps toward initiating a memorial fund towards supporting future internship opportunities. We haven't quite figured this out yet, but I hope that you will all consider donating a little something in honor of Mike when we do that, to help future groups of interns have the same experiences that you were able to have this summer, and expand that. All right, so our next step is, if you did an internship, I know Haley's told you about this, you need to do a report. So we'll send out a Google form that has pretty straightforward questions. We are responsible to NOAA, right? So NOAA gives us money, and we then in turn have to tell NOAA exactly what we did with it and explain to NOAA why we should keep getting this money. And so the more that you can tell us about how the internship shaped your understanding of environmental stewardship, or not, that this is a good field that you want to be in, or understand how your place within it might be something that makes sense to you. So we would really appreciate your feedback and all of that, and we share it. And before I move on, I just want to say that we need a really big shout out then to Haley Motley, who's our admin for Michigan Sea Grant. And she has been coordinating everything and doing all of your luncheons this summer and making sure everyone had a place and a supervisor that was responsive and all of that. And Haley has done an amazing job with that. And she just started in January. So really, she has hit the ground running, and we're really proud of her, as we are of all of you. So anyway, I just want to say thank you all for being here, for participating in this internship, for coming and sharing with us about what you learned and about with your families and your support people. And you really represent the best of what Michigan Sea Grant and what Michigan has to offer. And with you all as our, next generation of environmental stewards, I'm feeling pretty good about the future. So thank you all so much. All right, and now we're going to get started with our presentations for today. So each of you will come up and give your presentation 5 to 10 minutes, and then we'll have questions. And so there'll be a Q&A format after that. Again, thank you all so much for joining us today. We're so happy to have you. And Tyler is the first presenter. So why don't you come on up, Tyler?
Hi, everyone. My name is Tyler Bush. I'm a student here at the University of Michigan, studying Earth Environmental Science. This summer, I had the opportunity to partner with the Michigan DNR in an effort to understand the extent of user-created trails in the sand dune ecosystems of Lake Michigan State Parks. So throughout my presentation, I'm going to talk about kind of the background of my project, the goals that I had, the process that I took, the results I found, and then next steps moving forward. So my project is all about learning about user-created trails. I'll also call them social trails and informal trails throughout the presentation, all the same thing. These are trails created by people that are just hiking or recreating through an area like a park. These aren't designated or official trails in any way. They're just created and just go random places. So while the DNR does have all their official trails really well mapped with GPS and everything like that, they don't have any mapping of their social trails throughout their park. So we don't really know how wide they spread or where they go, which can be an issue. The reason this is an issue is because these trails actually cause a lot of problems. They're created usually as a method of convenience. They're shortcuts. They're often trying to get beach access or going to like pretty places in the park you can't otherwise access through official trails. These are usually unsustainable. They go through growing areas, vegetation. They really have no regard to the environment. Usually they're created unknowingly or without care. And when there's a lot of these trails in one spot, it can lead to a lack of clarity. It can cause people to become confused and then go on the wrong trail even more often. And all three of those combined are just a really big problem for dunes, because as more and more trails cut through, it destroys the dune grass that kind of holds in all the sand and keeps the dune in place. So I conducted my research at 7 different state parks along the coast of Lake Michigan, everywhere from Ludington down to Saugatuck, all the way down to Warren Dunes. So kind of just all across Lower Peninsula. I had like 3 main goals or questions I was trying to answer throughout this project. The first was, how far do these social trails actually extend? Like, where are they? How many do we have? How long are they? That kind of idea. I then wanted to find out how these trails actually impact the dunes and the surrounding environment. Do they have an impact? How important are they? What problems are they causing? And then lastly, how can we mitigate and reverse the impacts of these trails? So the way I did this, I used Field Maps ArcGIS. It's an app that allows you to use GIS and kind of both look at aerial imagery for an area as well as map it out using GPS. So the first thing I did was, at home, I would look at aerial pictures, kind of like what you'd see on Google Maps, Google Earth, of each park. And from there, I'd look at what trails I can see from above. I'd map those out. And then once I did that, I would actually physically go into each park and I would map these trails on foot. So I look at trails that I couldn't see from above, whether it be like hidden by trees or plants or something else. And then I'd also ground truth and verify all the trails that I found at home. After I did this, I looked at all my findings and I determined kind of what the main issues were in each park, where all these trails were, what kind of impacts they were having. And then I met with each supervisor from each park to discuss what I found and what possible solutions could be. This is just another look at what I was using. Picture on the right there, you can see is kind of what I was looking at both at home and while I was in the field. Those green lines are the social trails that I mapped out, whereas there's a small blue dash line you can kind of see in there, and that's a official trail mapped by the DNR. So results. So all the values listed down here, the miles, those are miles of social trails that I mapped. Those don't include the official trails in each park. So the first smallest park was Van Buren, 4.2 miles. Then I went to PJ Hoffmaster, which had a little under 13 miles. Grand Mere had a little over 13 miles. Again, if you missed it earlier, the green trails here are the social trails that aren't supposed to be there, and the blue trails are the official ones. So in each park, a lot more unofficial trails than official ones, oftentimes anywhere from like 2 times as many trails to like even 3 times as many. Then the next park was Warren Dunes with over 16 miles of unofficial trails, and then Saugatuck Dunes with just over 19 miles of unofficial trails. And then the 2 biggest ones I encountered were Muskegon, that had just under 20 miles of unofficial trails, and Ludington, which had over 30. So looking at this kind of like all in all, the big picture, there's 116 miles of total unofficial trails. So straight line distance, put in perspective, if you were to start the big house and walk in a straight line, you get to my hometown of Holland, which is all the way on the west side of the state. So definitely not an insignificant amount of trails. And then another visual, I estimated that I walked about 275 miles total between official and unofficial trails. So that would take you from the big house all the way to the Upper Peninsula. On average, there's about 16 miles in each park of unofficial trails. The lowest mileage being at Van Buren, which was the smallest park that I went to, so it makes sense. And the highest being at Ludington, which had just over 30 miles. So with this said, there are solutions to all these trails and the impact they're having. One of these is informative signs. These are signs that are put around parking lots or just entrances to trailheads. These include information about the dune ecosystem. They discuss good and bad practices about hiking, like leaving no trace. They stress the importance of staying on the trail. And then they also have maps to help people know where they want to go, and how far they have to go. That way they're not going off the trail because they're not sure where to go. Another way to help people out is with trail markers. These are markers that are anywhere from an actual map with numbers on it to just an arrow pointing you along the trail which way to go. And then another thing I found, all all the pictures in this are from different parks I was at. You're using branches or stones or fences to kind of keep people on trail and help in their mind verify that they're on an official trail. Another solution to people going off trail is with restrictive signs. So actually telling people, hey, you can't go this way, please stay on the trail, whether that be with this picture here on the left, or some people just not to enter. The picture on the right I found is the best picture. That's taken from a site of a national park. The visual on it is the best part because at a quick glance, you can see I'm not supposed to be here. I'm not supposed to go this way. And then if there's someone in the park that doesn't speak English or doesn't understand English, that can also help them, whereas a sign with just words on it isn't necessarily as clear. All these solutions so far have been kind of preventative measures, whereas actually replanting and transplanting dune grass is something that you can do to help reverse the damage that's already been done. So the first step is determining areas most deprived of the dune grass and the worst dune blowouts. Dune blowout is a kind of a hollowing of a dune. You kind of see on the image to the left here, this bowl-shaped kind of depression in the dune. That's caused by erosion, and over time, the stress of slope causes more and more dune grass to kind of fall away. And this is a big issue for dunes. In some areas, it's natural. We want to keep that, but in other areas, it's human caused, and we need to stabilize those dunes. So to do this, the marram grass, which is the primary native beach grass in West Michigan, can be replanted just by taking a shovel, digging it out a clump in a more stable area, and then replanting that to an unstable area, and it'll actually spread out and grow. So you can kind of make more grass. And then just monitoring that as you go and making sure nothing becomes overstabilized or understabilized. So over time, throughout all these parks, I kind of learned just how important it is as far as like good trail management, staying on the trails. And this project was really only the first step for these parks in the campaign against, dune impacts, dune erosion. With the information that I found, the park managers and park supervisors are going to be kind of taking next steps as far as implementing these signs, figuring out what trails to keep, what trails to block off, and hopefully kind of help grow the dunes back to what they used to be and keep the park accessible to everyone while also kind of helping save our natural resources. And then before I finish, just want to give a big thank you to Alicia Inkin, Jason Fleming, and Ray Falsing, and the rest of the Michigan DNR team. They were just absolutely helpful to me throughout the summer. Without them, I couldn't have done this project. There's many times that I had so many questions and had so many issues, and they're just right there to help me out. And then I just want to thank everyone for coming today to listen to what I have to say.
Yeah, I'm curious, are there any factors that you noticed that sort of contributed to more social trails being present in certain areas?
Like the call out and repeat idea.
Yeah, so the main things I found, a lot of times there's a lot of trails closer to the beach itself. So people trying to find a way to get to the beach that's faster than just taking an official trail there, or when there just wasn't an official trail going towards the beach, there's just lots of random small trails heading that way and then like kind of branching off of that. So that was like a main factor that I found.
Hi there, and this is Newell, and I hope you can hear me. First of all, I just want to say that was a great presentation and a really cool project, and I hope that you feel really good about the work that you did and the literal direct impact you're going to have on dune management and policy within these parks. Like, that's a really big deal, so you should feel proud of yourself. I got two questions for you. One is, how long did it take you to track like an individual park? Because it sounds like you were spent like days traipsing around these places. And the second question is, now that you've been a part of sort of the management of a park like this, how does that impact how you see yourself going forward? Like, did this impact your career aspirations at all, or just sort of on the track, but the
So each park obviously differed in size, so that had a big impact on it, as well as how many trails were in each park. Between using just like pictures from home that I could map out in a very short amount of time and actually walking the trails, each park took anywhere from like 2 days to 5 days to do. At Ludington specifically, I actually camped out overnight so that I could hike for longer each day, especially because it was just a further drive for me. So definitely depend on each park, but it was a lot of very physical days. And as far as my career path, I don't know that this has significantly altered what I want to do, but it definitely is an option for me going forward as something to consider. If what I decide to do doesn't end up working out. And I think working for the DNR, I want to be a volcanologist, so studying volcanoes. I want to go to grad school for that. And there is a way to kind of tie in coastal geology, then I would love to do that. But for right now, it's kind of still my focus is just focus on volcanoes. But dunes and coastal geology are really fascinating. So if I can find a way to tie them together, that'd be awesome.
Well, thanks for that. That was fantastic. I just want to make a quick note. Just last night, we were chatting with some of the facilities folks here at SEAS, and they said that in their little urban forest called Saginaw Forest here in Ann Arbor, they have exactly the same problems with people running off trails and having dogs off trails. And they said, we would love to have somebody do this kind of work in Saginaw Forest. So if you ever wind up at SEAS as a grad student, if you know anybody who's heading here and are interested in this kind of project, SEAS would love to have some folks do mapping and some signage design to try to figure out how to help people respect the trails. So thank you.
Okay, hi, my name is Hadley Van Vussy, and I'm going to do my presentation on the movement ecology of lake trout. So today we're going to go through some backgrounds, the methods, and the results of the project, and then a quick summary. So to get into it, this summer I did my project on the movement ecology of lake trout in northeastern Lake Michigan. I was looking at their broad scale movement within the lake and using acoustic telemetry. So that's like kind of the basis of my project. So some background about what is acoustic telemetry and why did we choose lake trout for our study species. So lake trout or acoustic telemetry is actually becoming a common method to look at the broad scale behavior, habitat preference, and then population dynamics of fish. And there's kind of two parts of acoustic telemetry. There's the tag and the receiver. So the tag can be surgically implanted into the fish internally or externally. And once that's inserted, when the fish is swimming around, the receiver under the water picks up the unique signal that this transmitter is giving off. And that receiver then picks up a GPS location. So then we can see this fish kind of moving in real time with quotation marks where this fish is moving within the lake. And the reason why we decided to choose lake trout as our study species is because in the Great Lakes, or specifically Lake Michigan, this species is under intense fisheries management because of their low numbers in the past. It's currently important to track commercial fisheries and recreational fishing. So the reason why in the past the numbers were so low, in the early 1900s, they were highly targeted for the commercial fishing industry. And then due to the basin-wide collapse in the mid-1900s due to the sea and expanding commercial fisheries direction, and then also contamination, their numbers got nearly to zero, which was pretty shocking, I think, to management in the Great Lakes. And so due to successful management, like wide lake stocking, their numbers were able to increase once again. So that's why they're kind of important to look at, because they don't have a self-sustaining wild population. They still rely on us to have a stable population. And then when I was looking at this project, I wanted to focus on three things, and that was mainly where are they going to be, so their location, how far are they going to travel, and the distance that they were going to travel, and then are they going to respond or go back to their spawning sites, so the fidelity that they have there. Okay, so the methods for my research. So we tagged 35 lake trout off of Good Harbor Reef, which is close to Leland, right off the coast there. And this is a spawning for these fish. So they were all tagged in October and November, which is the time that these fish spawn. And then the tags were surgically implanted into the fish by scientists on research vessels. And then our data was collected using an array of receivers, specifically using Great Lake Telemetry Observation System, or GLaDOS for short, which is a database that we can use that allows researchers in the wide Great Lakes Lake Basin to share the receiver data. That's pretty cool. Okay, so moving on to some results. But before we do that, there are some things that we have to consider when looking at this data. So our array in the Great Lakes, or specifically in Lake Michigan I'm talking about, is pretty small. So it's limited. It's limited our ability to really track the movement of these fish in the entire lake. So again, that's why our focus was on northeastern Lake Michigan. And we also don't have our full 2022 sample population back. So we believe that our sample population currently is about 30. And then I also wanted to show you a map of our receiver array. So this is Lake Michigan, and then those light gray crosses are where the receivers are located, and that star is Good Harbor Reef, and the fort coming off to the side is Grand Traverse Bay, to kind of orient you on this map a little bit here. So getting into the good stuff, this is our detection data. So you have the number of fish that were detected on the left, and then the months on the bottom. And so we can see that in October and November, which are those highlighted blue bars there, which indicates spawning season, that all 35 of our fish were tagged by November. And then as the water gets a little colder, we see that our detections decrease in February. We see our lowest number of detections. And then in May and June, our detections increase once again up to 30. And so the reason why we think we see this decrease in detections here is because these fish exited our array, or they're in areas of low probability of detection during this time. And so the reason I really want to show you this graph, because you can see that our sample population for our entire data set was not 35 fish all the time. We actually see a dip during the winter, and then we see stable 30 fish in May and June. And that's the timeline of our data. It's from the end of October to the beginning of June. So getting into my favorite part of my data. So this is our location data. This map shows the probability of detection organized by month. So the lighter the blue dot, the higher the probability of the fish being detected on that receiver. So what you can see here, and what's very interesting, is that these fish are kind of moving all over the place, and they're moving in all these different directions and in a very short amount of time, which is pretty cool. And then it's also important to note that these individual fish are crossing management zones while they're moving to these different places. So we know that from our data, they're moving north, they're moving south, they're moving to Grand Traverse Bay, or even kind of just staying on their reef. And what's interesting here is that Michigan kind of controls 8 zones in Lake Michigan. In our data specifically, we saw detections in MM3, 4, and 5. And what's important to note here is so these fish are crossing these management zones. Does that mean that we have to redraw these management lines? Do we need to reevaluate how we manage these zones? Because these fish are crossing not one or 2, or maybe they're even crossing 3 of these lines that we're seeing in our data. So that means that our harvest limits might be off, that our stocking estimates also maybe not quite right for these areas. So that's also important. And then some of you also may be wondering, why are these fish moving to these different places? So the short answer is we're not really sure based off this data, but we can speculate that prey distribution, prevailing currents, and water temperature have something to do with affecting their movement. But that hypothesis needs further exploration to really be answered. And then I'm going to show you a couple examples of how these individual fish are moving in northeastern Lake Michigan. So this is one individual fish. So that blue dot is the fish moving around in the area. Again, this is the probability of detection. Good Harbor Reef is where that fish started. And then we can see that it exits the array like in the south, and we lose this fish for basically the entire winter. And we see it come back into our array from the south again in May and June, and it's kind of hanging out in June in that Good Reef Harbor once again. And then, okay, so I was able to actually create some animations. Good Harbor Reef we can see here, and then goes into Grand Traverse Bay and is kind of staying there throughout the entire winter. We see that movement slow down when the water gets colder, and that activity will pick back up once again when the water gets warmer, and then eventually it will make its way back out into the Great Lake or into the Big Lake. So kind of cool there. So that was like kind of real-time movement of that fish. And then kind of just stayed on the reef. So we can see it's starting in Good Harbor Reef. It's kind of staying there after spawning season. It moves south off of the reef in November. Kind of doesn't fully exit our array and then moves back slowly to Good Harbor Reef. Kind of cool. So you can see like how these fish are moving in real time. I was able to create this using R. So this is a fish again. All these are starting in Good Harbor Reef, and then it quickly makes its way up into Lake Huron. So it's again, Good Harbor Reef is right off the coast of Leland, and it goes up through the Straits of Mackinac into Lake Huron and kind of hangs out there. We unfortunately lose it in March, so we have documented this fish from October to March. But pretty cool that fish was able to travel that far in that short amount of time. So now let's get into the distance data. So these are distance distributions shown by it's like the radius of the release where these fish were first released and tagged. So it's important to note that these distances are actually linear measurements. So they don't account for these fish moving around land masses, so they're very conservative. But we can also see that these fish, let me explain this once. So this is the number of fish detected here. So let's say like 5 fish were detected at in between 0 to 50 kilometers, and the max distance is on the bottom measured in kilometers. So what's interesting here is after spawning season in that November time, these fish start to move those larger distances, and then when the water gets colder months come in December and April, they're kind of staying in that 50-kilometer zone. And then when the water gets warmer once again in May and June, we're seeing these fish increase their distances, 2 fish even traveling 83 kilometers, which is pretty impressive here. And then also I wanted to note that these distances are only able to measure the distances in our array. So we don't know the fish that exited our array in the winter. We don't know how far they were able to travel. And then I also want to show you how these fish move in the water column. So this is a map of the min, max, and mean depth and temperature, depth measured in meters, temperature measured in Celsius. And the day of the year is on the bottom here. So those points are the mean, and the top and the bottom of those lines are the min and the max. So we saw, looking at this data, this is one individual fish, and looking at other fish's individual data, is that they kind of like to be in this 5 to 10 degrees Celsius range when they can. Obviously, in the winter time, they are pushed into colder waters. So they're not able to occupy that zone. And they're also making these huge vertical movements up and down in the water column day to day. So you can see that this fish started at 10 meters, moved all the way down to 60 meters in one day, or vice versa. So we can see this huge fluctuation in that they're occupying, but they're kind of staying in this relative about 5 degrees Celsius range. So that's one speculation we have of why these fish are doing this, is because they're following the thermocline, and they're trying to stay in this Goldilocks temperature zone that they like to be in, and they're making these broad up and down movements in the water column to feed. So pretty cool. So we got to look at their movements not only throughout the lake, but also in the water column too. And if you think back, that kind of got messed up. Think back to my objectives. I wanted to look at how these fish are spawning and do they come back to our sites or not. And unfortunately, we didn't get our 2022 data set back, so I could look at that. However, we did see in those other examples that I showed that these fish were starting to make their way back up to the reef by June. And the literature also shows that they have high fidelity to their spawning sites. So it's thought that these guys come back to their spawning zones. And kind of wrap things up, what I want you guys to take away from this presentation today is that these fish have ability to travel very large distances. They do it in a matter of months. And as they're moving around, they don't really have a distinct migration pattern after spawning. And as they're moving around, they're also crossing these different management zones. So maybe meaning that our harvest limits are off and our stock estimates aren't quite right for these zones. And I also was able to show you some general data about how these fish move in the water column. They're staying in around 5 to 10 degrees Celsius range. They're making these broad up and down movements, potentially to feed, but kind of day to day, they're making these large movements. And so this research is really important because by gathering information about how these fish move, we're able to really figure out how we can effectively manage this species. And also additionally, being able to learn more information about this species, we were able to potentially make decisions about how these fish are not only managed, but if we're deciding on a refuge site or drawing management zones, this data is important for that. And then I just want to say thank you for to the MDNR and Michigan Sea Grant for allowing me to have the opportunity to conduct my own research and work with amazing people. I also got to work on a lake sturgeon survey while I was there and the lake-wide assessment plan, or LWAP, with the Steelhead crew. And thank you for listening. And Ava Tackery and I have a blog post up if you guys want to read about our day on the Steelhead. And then a blog post about my research is also coming up soon.
Skill or a lesson that you picked up during your internship that you will be able to hear? Academic work or future careers?
So this is kind of my first time doing my own research and really working with R, which was a whole thing in itself. And I had Ben Tershak really help me with that. But I think I learned a lot and how it can help. All my data here is made in R, so I'm kind of excited that I have that new skill to show for my future employers.
Okay. Hi, everyone. I'm Delaney Saul. I'm a student at Calvin University in Grand Rapids, and I was doing my research on green collar jobs and adolescent mental well-being. So I was working with a nonprofit organization called Plaster Creek Stewards. Plaster Creek is a watershed and a creek, and it runs into the Grand River and then eventually into Lake Michigan. And the goal of Plaster Creek Stewards is to focus on restoring the relationship between people and land. Plaster Creek watershed is one of the most polluted watersheds in Michigan, and just faces a lot of environmental justice issues that our idea, I guess, that our organization is that's stemming from mainly this disconnect between people and the land and the way they're using the land. And Plaster Creek Stewards has a program called the Green Team, which is 2 3-week sessions of high school students that come and work with them, learn about watershed ecology, and learn about implementing green infrastructure solutions. So we install curb cut rain gardens, we install bioswales, that kind of thing. And then that's sort of coupled with classroom time. So I was working with the Green Team this summer and doing research on the impacts of this program on their mental health. The Green Team program is an example of what we call a green collar job. So a green collar job is just sort of doing hands-on work in the environment, that kind of thing. And there's a lot of research on how green collar jobs can be beneficial to mental well-being. So I was looking into that because there's a gap in the literature with regards to adolescents, and then also adolescents with intersecting identities. So I was trying to look into specifically sort of what perspectives on nature and what different identity factors sort of change this influence and impact it. So my methodology, I did pre and post surveys. So the surveys included a bunch of questions about the students' perspectives on nature. And then the Warwick Edinburgh Mental Wellbeing Scale, that's a well-being measurement that's been validated for adolescents, which is why I used it. And then some demographic questions. And then I also did pre and post interviews, and those were a little more in depth, sort of examining the students' stories behind their relationships with nature, their goals for their program, sort of what they learned and took away from the program, as well as any feedback they had for how to make it better. And then finally, a big part of what I was doing was just building a supportive and like caring mentorship relationship with the students. I was hanging out with them, every day for 3 weeks. So just, like being genuine and building relationships was fun and important, but I think also helped me get good data from the students. So I've completed my statistical analysis of the survey results. There were only 22 students, so there are some like more in-depth statistical analysis that I wish I could have done if I'd had more, but I was able to get some pretty reliable statistical analysis. So the first thing that I wanted to point out to you is the importance of school and family encouragement in developing a relationship with nature. So from my nature questions in the survey, I developed sort of an index that I'm referring to as cultural access right now. And it's basically the access that a student has to having a strong relationship with nature based on like if their school provides them with opportunities to be outside, if their parents encourage them to be outside, if their friends and family enjoy spending time outside, all of that kind of thing. And then also their entry well-being score. So I found that students that have a higher cultural access, so have more opportunities growing up to develop a relationship with nature and spend time outside, have a higher well-being score when they come into the program. And even after controlling for demographic variables, that was still true. So I think that, it was sort of just like affirming of the importance of having people that encourage you to develop a relationship with nature and the importance of being outside and like feeling connected to nature. So then I found also, which I think was especially interesting, that students that entered the program with a lower cultural access index score took more or had a higher change in their well-being from the program. So it was more impactful for students that came in with less sort of experience with nature beforehand. And then I found a few different trends in demographic variables. The first one is a gap in cultural access. So white students were more likely to have a higher cultural access score than BIPOC students, which I think could be for a variety of reasons. I'm sort of trying to sift through the literature right now, sort of going back to sort of figure out why that could be, but that is one trend that I saw. Also, there was LGBTQ plus students were less likely to have a high change in their well-being. So there was no significant like difference in how much their initial score was, but it was more likely to stay the same for LGBTQ plus students as opposed to non-LGBTQ plus students. And then finally, I found that for students that were assigned female at birth, their entry well-being score was significantly lower than that of students assigned male at birth. So then I'm also sort of in the preliminary process of analyzing the interviews. So I'm coding them and sort of going through that right now, but I am starting to sort of see some themes emerging. So the first one is engagement. A lot of the students talked about how valuable it felt to them to be able to do hands-on work with the environment and see a project from start to finish. There were a few where we would go to like a bioswale and it'd be like nothing. We plant, I don't even know, probably like 20 to 30 flats in a day. And it was just like really cool to see. And I think the students really found that valuable as well. And a lot of them mentioned that in their exit interview, as well as the sort of excitement that they had seeing projects that had been implemented 2 or 3 years ago and thinking like, wow, like that's what we did today is going to look like later, and really like feeling like they had made an impact on their place. And I think it's especially cool because the students that work for Green Team live in the Plaster Creek watershed. So this is sort of directly impacting their place that they inhabit. And then a lot of the students, almost every student actually, also mentioned just being really excited about all that they learned during the program. That's things about environmental justice and watershed ecology, but specifically a lot of them mentioned really enjoying learning the names of different native plant species and sort of like walking around with their friends and family and being able to point out different plants that they knew. So that was something that was really exciting to the students. And I think increased sort of their sense of confidence in themselves. Yeah. And then finally, a lot of them sort of weren't expecting this, but came away with a greater sense of belonging and sense of community and made some really good friends in the program. Most of them that mentioned this were pretty surprised about it, but I think that definitely had an impact on their well-being as well. So yeah, I think a few things that I want to emphasize from this research is how important the role of schools as well as family are in not only allowing students to develop a relationship with nature, but also in just how that relationship with nature and that encouragement can directly impact students' mental well-being, and how they feel about themselves and how they feel about interacting with others. So that's really important, as well as the influence of the Green Team program and other green collar work opportunities can be super important for mental well-being. And I think all but 3 of the 25 students had an increase in their mental well-being score. So it was definitely something that was very impactful for them. And then I also want to emphasize sort of the needs of marginalized students. A lot of marginalized identities would either have like less of an impact from the program or would come in with certain factors that might make it more difficult to like benefit from the program. So that's something that I think is really important and something that I'd like to investigate more, especially because I wasn't able to get into intersecting identities as much as I wanted just because of my smaller sample size. So yeah. And then I'd like to thank the Michigan Sea Grant for making this opportunity possible, as well as my supervisors and coworkers at Plaster Creek Stewards, because they're amazing and just made it really fun. So yeah.
I'm a Calvin alum and I worked a little bit with Plaster Creek Stewards and Dave Warner's and all those good people when I was there. So glad to see it continuing on. And speaking of continuing on, you said that there's some future avenues that you're hoping to pursue with kind of tying up the interviews and some more cultural analysis. What do you see as your continued participation with this work? And then where do you think future researchers, either interns like this or other folks at Calvin, will take it?
Yeah, so I think as far as sort of the future for this at Calvin, I'm hoping that the research that I did this summer will be really helpful in sort of fine-tuning different aspects of the Green Team program and making it as beneficial as possible for the students. This is something that I was really passionate about and just, yeah, kind of sad to see end like the time that I got to spend with the students. So I think it's definitely just like affirmed for me that this work is like so important and also that it's something that I'm like particularly passionate about.
2 things. So first, I want to say that I think your work is not only very cool, but very timely. Like if this is something that you want to pursue going forward and in graduate school, there are literally more opportunities for research like this and funding than we have people to review them. So this is a great place to expand into if you're thinking about an area like this for your career. But just a thought about your research for the summer, how much do you think that the increased sense of well-being in the students had to do with sort of forming a group and a bond of people who like happened to be spending time outside versus the green collar aspect of it? As in, like, could any summer camp that was spending outside, would you expect it to see the same results versus one that was really emphasized the relationship with nature and learning, you know, plants and stewardship?
Yeah, so I guess my best way to answer that question is to explain the well-being scale a little bit better. So it's an 18-question scale, but it's sort of divided into 4 different types of well-being. So there's sort of a sense of self-esteem, and then there's emotional well-being, so like a sense of being relaxed and calm and happy. And then there's relational well-being, so that's their interest in other people, and feeling close to other people. And then there's a cognitive well-being, which is like their ability to think, their ability to solve problems. So what was really interesting is for a lot of the students, even though they were building these close relationships, the sort of sphere of well-being that had the least change was relational. So that I don't really have the answers for right now, but I think what I'm thinking is that there is something more to this program than the friendships. I think maybe an outdoor program could do similar things, but I think a big part of it for them was the learning and like the ability to make an impact on their place and on their community. So yeah, I think there's something to that.
Okay, so I hope you enjoyed that little recap of what my summer was like as a research intern at Thunder Bay National Marine Sanctuary in Alpena, Michigan. And I figured I'd start off by introducing myself. So my name is Madeline Sadler, and I am a third-year undergraduate student studying environmental science at Northern Michigan University in Marquette, Michigan. And I was first introduced to the Thunder Bay National Marine Sanctuary because I was fortunate enough to grow up in Alpena, Michigan, which is where the sanctuary is. So we would take field trips in elementary school to the sanctuary. I ended up getting scuba certified, diving shipwrecks in the sanctuary. And just after graduating high school, I actually worked as a deckhand and tour guide on the sanctuary's shipwreck tour boat as well. So this summer, I was super excited to have this opportunity with Michigan Sea Grant to further my role at the sanctuary, studying freshwater acidification at the Thunder Bay National Marine Sanctuary. So the formal title of this project is Freshwater Acidification, Monitoring and Environmental Stewardship in the Great Lakes, and it is a partnership project between the Thunder Bay National Marine Sanctuary, the Great Lakes Environmental Research Lab, which is right here in Ann Arbor, and NOAA's Ocean Acidification Program. So the overall goal of this program, or this research project, is to gather baseline data and establish the first long-term monitoring system for freshwater acidification in the Great Lakes. So the process of acidification starts with a gas called carbon dioxide, and this is emitted into the atmosphere naturally through processes like plant and animal respiration. And we call this normal carbon dioxide. But on the other hand, rampant carbon dioxide is carbon dioxide that's emitted into the atmosphere in harmful amounts through the burning of fossil fuels. So once in the atmosphere, it creates a heat trapping blanket, which causes average atmospheric and water temperatures to rise. And water bodies like Huron absorb this atmospheric carbon dioxide, and a chemical reaction takes place that creates carbonic acid. So this lowers the water's alkalinity or its resistance to acidity, and also lowers the water's pH, which makes it more acidic. So an increase in acidity can cause problems for fish populations. It can inhibit their ability to perform their basic activities like eating, growing, and reproducing. And it can also really affect the lower food chain populations of the Great Lakes. It can also increase the probability and growth of harmful algal blooms, which are toxic to not only humans, but also aquatic species and other fish populations as well. Acidification is often referred to as osteoporosis of the sea because it reduces the amount of carbonate available in water, which is essential for calcifying organisms such as this pteropod I have up here on the screen to create and sustain their durable shells. So for this project, we chose 6 sites within the boundaries of Thunder Bay National Marine Sanctuary, and the sanctuary spans just over 4,300 miles of protected waters. And within those 4,300 miles are what are believed to be 200 shipwrecks. 100 of those 200 are fully identified and documented. So we chose our 6 sites not only because they are well marked with what's called buoys, and they're super easy to return to, but also because of their differences from one another. So some of our sites were near shore, and some were out in open water. Some of them were shipwreck sites and others weren't. And some were in really deep water and others super shallow. So the idea behind doing this is that we'll be able to tell how different environments are kind of impacting the presence of acidification. And thinking long-term in the future, we may also be able to tell how acidification is impacting the sanctuary's cultural resource, which is the shipwrecks. So my role as an intern was to tackle the surface sampling portion of this project. So I was tasked with visiting each of those 6 sites once every 2 weeks. And I did so via research vessel, paddleboard, and kayak. And I had to gather 3 samples at each site, once every 2 weeks, using what's called a peristaltic pump. So that is that big yellow box that you see in each of those photos. And essentially it's designed to draw water from about 1 meter below the surface through the tube and into my sampling vials. Once the samples are gathered, they are then sent down to the Great Lakes Environmental Research Lab here in Ann Arbor, where project scientist Dr. Reagan Herrera processes them for measurements of pH, total alkalinity, total carbon, and dissolved organic carbon. And those are water chemistry indicators that will be able to tell us how climate change and acidification are affecting the Great Lakes. So we haven't had that data returned to us yet. So unfortunately, I don't have any solid answers for this year's sample season. Thunder Bay National Marine Sanctuary, one of their main priorities is providing students, educators, and the general public with the resources and opportunities to expand their knowledge through experiential and exploratory learning. So as I mentioned earlier, I worked as a deckhand on the Lady Michigan, which is a 65-foot glass bottom tour boat where passengers are able to get an up-close view of some of the sanctuary shipwrecks. So this summer, I spent some time on board presenting about freshwater acidification. So I gave just a brief kind of overview of what the process actually looks like, and then followed it up with a little demonstration about sampling processes. And an example of this super cool experience that I had pretty early on in this project, back in May, a student group from Stockbridge High School visited the sanctuary to deploy like an autonomous mini sailboat that they created, which was super cool. So after they deployed their sailboat off of the stern of the Lady Michigan, they then turned around and helped me with the acidification sampling processes, learning how to use the peristaltic pump as well as a CTD sensor. So that was a super cool opportunity as well. And I'd like to wrap up by saying that although my time dedicated to this project is coming to a close, the research is far from over. For a number of years, the Thunder Bay National Marine Sanctuary was the first and only freshwater marine sanctuary. Within the past couple of years, we were joined by the Wisconsin Shipwreck Coast National Marine Sanctuary in Lake Michigan, and more recently, 2 other sanctuaries in Erie and Ontario were also proposed. So the overall goal of this project is to really expand and share this monitoring system with the other proposed marine sanctuaries as well. So in the grand scheme of things, the work has just begun. Thank you.
What's a skill or a lesson that you picked up this summer that you're looking forward to carrying into the future for yourself?
So I would say, as far as skills go, that I will take with me and into my future career would be communication skills with people of all ages and all abilities as well. I haven't always been around kids up until this summer, I would say. So I really got to kind of learn the ways to effectively communicate to people of all ages, including kids. So that was super huge for me.
And speaking of communicating, I'll take this as an opportunity to make a plug for the blog. So I am the editor of our fellowship and internship blog, and it is a great way to stretch those communication skills by writing something about your summer experience. That can be anything from kind of a summary of your whole time. It can be a cool day, an interesting person you met, a tool you learned, lesson, something you picked up, takeaways, advice for future interns. And it's a great way to showcase those communication skills that you have, or to grow some fresh ones and to show that to future supervisors, employees, academic advisors. It's a really great resource. Any other questions?
See, sinkholes in the news a lot lately. That site just for spatial reasons, or was it something, a lot of certifications going on there that like,
Yes, that's a good question. So the Middle Island sinkhole was one of our 6 sampling sites, and that has drawn researchers from all over the world within the past couple of years. So we kind of chose that site because of its unique microbial environment to kind of see how that's affecting acidification, but also because it is out in open water and it's a super well-marked place as well. So there's potential for the unique environment that exists there to affect acidification. We just don't know that yet, but
Hi everyone, I'm Ava. I'm from the University of Michigan, and this summer I had the pleasure of working up in Charlevoix at the DNR Fisheries Research Station on a whitefish river spawning project. So this is just kind of an overview of what we're going to be going through. But to just give a general introduction to the project, these were all of the lovely partners that I worked with. Throughout this project, it was a super collaborative effort, which was an amazing experience with the Nature Conservancy, the Wisconsin and Michigan DNR, and also the Sioux Tribe and the Little Traverse Bay Band. So our overall research objectives were, first, collaboration. We wanted to make sure that this was a collaborative effort and that all of the involved partners were super heavily involved. We also wanted to expand the scope of the survey and try to study as many rivers as we could throughout this short 3 months over the summer. And we also wanted to generate some kind of an end product that is accessible to whatever agencies might want to use this data for restoration in the future. So why should we care about whitefish in the first place? In the 1800s, Lake Whitefish historically spawned upstream in Lake Michigan tributaries, and then the logging and damming industries began. And that was super detrimental to whitefish spawning habitat. And there was a whole bunch of other issues that came within the last century too, like invasive species such as sea lamprey and zebra mussels, and also intensification of climate change and shifting stratification of the lake. So all of this has degraded spawning habitat significantly, and lake whitefish are pretty limited to just spawning on reefs now. There's not a whole lot of tributary spawning that happens anymore. But lake whitefish are a super important fish culturally to tribal and commercial fisheries. They're really important, but recently in Green Bay in Wisconsin, there's been some novel whitefish spawning in tributaries there. So we're particularly interested in that, in how that could translate to reestablishing runs and other tributaries in Lake Michigan. So this is kind of a little overview of the whitefish population over time, just to show some key points. The most concerning thing is recently, on the far end of the scale, how much it's declined in the past 20 years or so. So that's what we're concerned with right now. But this is just going to be an overview of our survey area that we chose and specific methods. So we had some priority rivers that we chose throughout both the, sorry, I gotta go through these. It didn't update, so I wasn't able to change this. Okay, so these are all the rivers that we wanted to focus on, and we were able to get to most of them. There were some that had dams pretty far close to the mouth, and those maybe were less priority for us to study because going forward, if we were to reestablish some of these spawning runs, the dams would block fish passage pretty far upstream. So those aren't as important to the study right now. But we wanted to access some tributaries that fed into these rivers too, like smaller creeks and streams. So we were able to get to quite a few of these over the summer. So first, I would start by going on Google Earth and flipping through or panning through these rivers and looking for these riffle areas. I don't know if you can tell based on this picture, but any kind of riffly white disturbances, I would zoom in and try to pinpoint that area and be like, okay, that is somewhere that I want to physically go in the field and see if the rock habitat and the bottom composition of the river matches up with what I'm seeing remotely. So then we would go in the field and take pictures and collect a lot of data based on the vegetation, the sand, what kind of substrates there were, and I would log that all in a GIS program. So that was kind of the steps I would take to make this large database of the river segments. So this kind of turned into a big map in the end. And some of these I would go out to the river and pick certain spots based on where I identified remotely. And I would pick these spots, access them, take photos, take the data. And then other rivers, I would take a canoe out and actually go float through that river if there was a lot of tree canopy that blocked the remote imagery. And I wanted to make sure that I didn't miss any good spots. So I went through and logged the substrate, which is just the rock bottom composition, how much vegetation cover there is. When whitefish spawn, they pretty much just rely on the rocks already being stacked up a certain way so their eggs can fall into the cracks in the rocks and be protected throughout their incubation period. So we don't want a lot of sand coming in and filling in those crevices. We want them pretty clean. So that was what I was looking for, and I made this map layer on GIS. So why is this database going to be important going forward? In the future, we really want to make this information accessible to agencies who might want to use it for restoration or reestablishing spawning runs. In the past winter, in the Carp River, which is up in the UP, some of our project partners were able to do some experimental egg incubation where they put eggs in a certain spot in the Carp River and let them sit throughout the winter incubation period, and then measured the hatching success rate in the spring. And that was really interesting because it's kind of just the start of this project and where this can go in the future and how this can translate across different rivers. And then earlier in the summer, when I first came up here, I was able to participate in environmental fry stocking in the Pine River, which is kind of near where the Carp River is. We stocked about 2,500 fish in the Pine River, and they were a couple inches long. They're a little big. And then we tracked their movement upstream, downstream to see, are these fish hanging out in the river? Are they going to imprint, or are they not interested in staying in this river and they just want to move right out into the lake? And even a couple weeks after we stopped fish, we were able to recapture a couple of them, which is really impressive because we didn't stock that many. So this could be a really cool option in the future, using the rivers that I survey to start trying to reestablish these runs in other rivers in Lake Michigan. So outside of this project, I was able to participate in a lot of other work up at the station, including the Steelhead survey. I was also able to do a lot of work with the Chinook stocking at our weir, and some larval sampling early. And then also just a lot of outreach with the local elementary students and summer camps, which is really cool. So acknowledging all the amazing partners that I was able to work with, I'd like to thank everyone. It was an amazing opportunity. So enough for now. Thank you.
So working with fisheries, did this play into your current career hopes or change anything? How does this fit into where you see yourself heading?
So I feel like before the summer, I really never had any full exposure to the fisheries field before. I don't even think I really knew all that it entailed until getting to pretty much get plunged full forward into this fisheries project. And I think it's really changed the way that I'm envisioning my career path, because I don't think I really have a set career that I'm like entirely nailed down on. I think I've just been jumping back and forth between a lot of different fields, but I really like just the field work itself and just the schedule and like just being able to work outside all the time. And whether or not I end up in fisheries, I know that working on the water is something that I really enjoy and something that I see myself continuing in.
So again, this is a great area to go into. Just saying, there are a lot of jobs right now based on some political and funding factors. In the past, there aren't as many people doing fish ecology, and there are a bunch of people getting close to retirement. So like, great career opportunity if you decide you want to do this and stay in the Great Lakes area. I also wanted to ask a little bit about your like field safety practices. So can you talk to me about, when you're going out? I mean, I assume when you're sitting at home with GIS, you're by yourself, right? But like when you're going out in a canoe, did you go out as teams? Like, talk to me a little bit about those practices.
Yeah, so on any average typical field day, I would never go out alone. I'm always at least with one other person, usually a couple. If we're floating in a canoe, we would never load it more than with 3 people, and then our equipment, which we didn't have that much equipment. I only had a couple measurement devices, so it was nothing bulky. Always PFDs, always wear life jackets. And we'd always bring an extra rope just in case, for some reason, if we encountered a section of the river that was too rapidy, we could tie ropes to the canoe, walk along the shoreline, pull the canoe over the rapids, and then get back in. And then, of course, we learned the hard way the importance of all of our belongings in the canoe, just in case there's some encountered obstacles. But yeah, anytime we were out hiking back in the woods, because all these access spots in the river were pretty far back, really important to wear protective clothing, because a lot of times you're just hiking through really thorny brush, and then just being with other people. I wouldn't do that alone. And then wading through the rivers too, it was really important to kind of scout them ahead of time and try to make sure that it's safe to cross and that there's not going to be any weird drop-offs. So all the parts of the river I was physically getting into, I made sure that I was like pretty comfortable walking across. And if at any point I was like, turning around. But yeah, thankfully there wasn't anything too crazy that I encountered this summer.
Hi, so my name is Raven McKechnie. I'm a student at Lake Superior State University. And my project was on determining the threshold level and sensitivity of eDNA testing for the freshwater invasive algae Didymosphena geminata. So a lot of people don't know about Didymo. One of the names for it, the common names, is very lovingly called rock snot. And it's because it looks kind of like snot and it sticks to rocks. But the harmful thing about this diatom is it forms really thick, dense nuisance mats, which in some studies have been found to be detrimental towards densities of benthic invertebrates. In some studies, especially in New Zealand where it was first detected, they found that it has an economic impact as well on the fishing industry. So overall, not great. It was detected in Michigan in the St. Mary's River in 2015. And since then, it has only increased. In the past few years, it was also detected in the Upper Manistee River and the Boardman River as well. Unfortunately, the only way to stop the spread of Didymo and the mats that it creates is to be able to detect it to prevent it from going into other inland bodies of water. Fortunately, there's not a lot of methods for detection, so that's where my project comes in, and that's where eDNA comes in. So eDNA is environmental DNA. So essentially you can take a sample from soil, air, or water, and then use that to detect whatever it is you're looking for. So in my case, I'm trying to detect Didymo, so I have to take water samples. So the objective of my study in particular, since eDNA has been used a lot for fish very successfully, but not so much for diatoms in particular. So the objective of my study was to figure out the threshold and sensitivity levels. So essentially I'm trying to figure out the threshold and the amount of density you need in order to be able to detect it using eDNA, and also the sensitivity, trying to figure out how far away I can detect it from a known infestation source. So to do that, for the sensitivity, I am sampling from a known infestation in the same areas. I have 3 sample sites, which is 0 meters, 500 meters, and 1,000 meters away from the known infestation. At each of these sites, I'm also trying to see if I take an increased volume of water. So I have 10,000 liters of water that I'm filtering and 30,000 liters of water that I'm filtering. I'm trying to see if I increase that volume, if it kind of boosts the concentration that results as well, so that can help with sampling techniques. And at each of these sites, I have replicates for each of the volumes at each of the sites for 18 total samples. And in order to sample, I use a nested plankton drift net. Goes through an initial mesh just to get rid of a lot of the stuff that we're not looking for. And then it goes through a 30 micron mesh, and that's around the size of the cell that we're looking for. Ends up in a 250 bottle, and then from there we do DNA testing. For the threshold, I use an experimental streamway system. So I have 16 raceways, which each have different treatments. I have 4 treatments. I have my control. I have a high density, a medium density, and a low density, trying to just see how the concentrations differ at the end. And this is kind of a brief overview of the analysis because there's a lot of steps to it. The bottom column is mostly just for the stream table and density measurements, trying to figure out the total algal biomass and the density of the algae that we have on the rocks. And then the top is kind of a very simplified process for DNA extraction, and then dPCR, which is what I use for my eDNA process, and then ends up with data analysis. So the results. Unfortunately, the stream table experiment didn't end up working out for a variety of reasons. When we ran the concentrations, nothing was coming up, and it's assumed that based on what I know, it just didn't meet the threshold level for detection. But we do have a lot of interesting results from the St. Mary's. I did a pilot study just to see how this works and if it would work in October. And this was kind of some of the data that I got from my pilot study. These are box plots that I have comparing the concentration in copies per mil versus the distance away from the known source, which, as you can see, there's not a significant difference between how far I am away and concentrations that vary. And versus this time around in June when I did it, there is a significant difference in concentration versus the distance that I'm sampling. But when you take into account the amount of liters, considering that there's 10,000 and 30,000 liters, it's not a significant difference. So what this goes to show is that it doesn't necessarily matter particularly how far away you are, at least for me, from 0 to 1,000 meters, or increasing the volume wouldn't significantly change the difference, which is important because the amount of sampling time for 10,000 and 30,000 liters is 3 times as much. So it can definitely change the efficiency if you are sampling out in the fields. So what's next? I have to analyze my microscopy and chlorophyll A data from the stream table experiment. I also plan on redoing the stream table experiment in the fall. Unfortunately, Didymo is only a fall and summer spawner, so I kind of have to wait in order to get rocks for the stream table. And then the things that I'm definitely going to change during the stream table experiment when I rerun it is I'm going to decrease the velocity of flow. I'm increasing the amount of liter samples. I'm also going to establish positive control just so I can know that it's working this time. And then some people that I would like to acknowledge. I was able to do this project from funding from the Michigan Invasive Species Grant and obviously the Michigan Sea Grant. I would also like to thank all of these people that helped me during my sampling events. Especially the stream table one is very labor intensive. And then I'd also like to give a big thank you to Shawnee McMillan and Dr. Murky because they mentored me through this whole project and were a really big help. So thank you. Any questions?
How does this field work feel for a future career track? Is this something you can see yourself doing in the future?
So I am actually an animal biology and pre-vet major. So this is a little bit outside of my fieldwork. But the reason why I kind of got into this research is because last year I was able to work for my tribe, the Sioux Tribe of Chippewa Indians, environmental department as an invasive species intern. And so I have a very big passion for, even though this is technically a nuisance species, I have a very big passion for invasive species, things that can harm the environment, and what I can do to kind of help. And when I started working at the Center for Freshwater Research and Education, me and my mentor, she was the one that interviewed me for the position, and this was to be a 15-minute interview. We ended up talking for 45 minutes all about Didymo and everything about it. And so it kind of just spurred from that as like a nice passion project, something I'm passionate about, and something that definitely Lake State's very passionate about as well.
I was just wondering what went wrong with your stream table, you say?
So in terms for the stream table, when I say threshold of detection, I guess I didn't mention it. When we're taking the water samples for the stream table, it is the same method as when we're taking them in the St. Mary's, but we're only taking 10 and 30 liters, which compared to 10,000 and 30,000, that's 3 volumes of power. Obviously a very big difference. And when you go and compare the concentrations that we have, even at 10,000 and 30,000, it's not a lot, especially for, in terms of detection level, it's 2 cells per mil, which in terms of taking 10-liter samples and 30-liter samples, it just wasn't, from what we theorized, enough water. And also we had a very high velocity of flow, which was fighting against us for our sampling. And then on top of that, we have a very extensive piping system for the stream table. So by the time everything works its way through the net, there's just not probably a lot of cells that get sloughed off for detection. So we just weren't able to detect it this time. But I think given everything we're changing for the next time we do it, it hopefully will work. We're taking a much bigger volume of water this time.
Right. Hello, everyone. My name is Kyla Charlebois. I'm a sophomore at the University of Detroit Mercy. I've been working on this project alongside my advisor, Dr. Nutu, and we have been researching the occurrence of endocrine disrupting compounds, pharmaceuticals, and personal care products upstream and downstream of an urban wastewater treatment plant. So to begin, the Great Lakes Basin contains approximately 84% of North America's surface water, supporting 9 major metropolitan centers and more than 30 million people. These metropolitan centers and urban watershed are profoundly influenced by human activity and are heavily developed. The quality of urban surface waters, as the Detroit River, has become an emerging concern due to the increased detection of pharmaceuticals and personal care products, pesticides, and endocrine disrupting compounds. Pharmaceuticals and personal care products are defined as any chemical compound and its constitute that's used for personal health, cosmetic reasons, or to enhance growth and health of livestock. And then chemicals that can disrupt endocrine-related processes are known as endocrine disrupting compounds. In urban environments, these chemical compounds are released from municipal wastewater treatment plants, sewer overflows, and non-point sources such as groundwater infiltration and runoff from agricultural, residential, and military sites. The consequence for the emerging concerns at low levels remains poorly understood and is therefore one of the current emerging issues within the Detroit River. Moreover, little is known regarding the spatial distribution of these products, so it is unclear whether these compounds are being distributed just a few meters from Detroit's wastewater treatment plant, or if these compounds are being widely contaminated and contaminating surface drinking water, which can be both a human and ecological health concern. So measuring the pharmaceutical and personal care product, pesticides, and endocrine disrupting levels is important to determine at-risk watershed, which can be used as a benchmark for future remediation efforts. So water samples were collected from 5 locations in the Detroit River watershed. So 2 samples were collected from upstream of Detroit's wastewater treatment plant and 3 samples from downstream of Detroit's wastewater treatment plant before discharging into the Detroit River. The water sampling locations were chosen based on proximity to water management facilities, as well as importance for recreation and wildlife habitat. So the 2 upstream samples were intended to provide a snapshot of the total pharmaceuticals and personal care products, pesticides, and endocrine disrupting compounds present in the water before being treated. And then the treated wastewater was intended to provide a snapshot of which pharmaceuticals and personal care products and endocrine disrupting compounds are present after being treated that may make their way into the Detroit River watershed at detectable concentrations. The sampling kits contained amber glass bottles for both the pharmaceutical and personal care product, as well as the endocrine compound disrupting analysis. The water samples were collected from sites 4 to 6 off of a boat using a 2-meter polyethylene dipper, and then from sites 1 to 3 from the shore. All gloves were changed between each sites, and then equipment was all disinfected to avoid contamination. All the samples were then kept at 4 degrees Celsius prior to arriving at commercial laboratories for analysis. At each sampling location, the water depth, water temperature, water pH, total dissolved solid, and water conductivity was also recorded. And then the analysis for the pharmaceuticals and personal care products, pesticides, and endocrine disrupting compounds was performed in the lab using liquid chromatography and tandem mass spectrometry in both positive and negative ion mode. So in terms of results, we are still running the analysis and waiting to receive those results from the 2 commercial laboratories. We are trying to analyze the detection of these compounds from the influent and effluent sampling of the wastewater treatment plant to determine which is more prone to contamination. Some of the substances we are interested in looking at include sucralose, sulfamethoxyl, acetaminophen, lidocaine, atrazine, caffeine, and many others. Of those 17 target substances, we want to know if they'll be detected in all sampling locations, or if at least one of those substances is detected in one sampling location. These concentrations will be measured in part per billion and part per trillion, and this will allow us to see if these concentrations of these compounds have reached or will be close to reaching concerning levels for the 4.5 million residents of both Detroit, Michigan and Windsor, Ontario, Canada that rely on the Detroit River as their main water drinking source. So in summary, we'll be able to determine whether the target compounds are more concentrated in the influent or effluent sampling in close proximity to Detroit wastewater treatment plant. Reevaluating the findings during the fall season and then comparing them to what was found now in the summer season is important and will be conducted in our study. All results of the study will also be published in our research paper. However, future studies must continue to evaluate the human, animal, and ecological health impacts relevant to the exposure of frequently detected compounds in the Detroit River watershed. Thank you.
So this is one of the emerging areas of concern for water quality all over the U.S. And so it's difficult to do these analyses, as we know, which is why they're sending them out to a commercial lab. And so it's exciting data. And so I'd like to ask about your continued role in this going forward. Have you talked to your PI? Are you planning to play a role in the data analysis and interpretation, or are you sort of moving on after the summer? Talk to me about your future with the project.
So once we get our results back from the first sampling, we will kind of take that and analyze them to see if we did get the results that we wanted or if not. And then from there, we'll basically come up with a new plan to do a second sampling in the fall season to compare those results, see how they differ. And then if we didn't get any results, we were going to come up with a new plan, try different locations to try and find these target substances.
So basically, my project was on environmental injustice, as you can see by the title. My name is Ariel. I'm from Detroit, Michigan, and I attend Michigan State University. I'm studying sustainable parks, recreation, and tourism with a minor in sociology and leadership in integrated learning. My career goals are primarily centered around community development and sustainability in impoverished communities. And this summer, I worked with the Detroit Zoological Society under the leadership of Mr. Mike Reed. So kind of before I begin, I want to go into like what environmental injustice is. As listed here, the definition is actions that harm the environment while alienating specific groups and communities. So it primarily impacts like impoverished and minority communities. These people, like the people in these communities, are often unaware of the unfair environmental conditions. So some of the examples here that I listed were like the Flint water crisis, manufacturing companies near homes and urban communities, and landfills near homes. So like I'm from Detroit, Michigan. So like if I'll drive down like to say like downtown, there's like a lot of factories like just there. Like we're known as the Motor City. So like it's just like a lot of factories like anywhere you go really, and like that polluted air is like what people are breathing in. That's really unhealthy. It's really dangerous. So some of the health risks that come with that are like, you know, asthma, lead poisoning, obesity, and some forms of cancer. And I also kind of like included a did you know slides. Like these are some fun facts that I found like while I was researching. studies have found that people of color are 47% more likely to live near hazardous waste than facilities than white Americans. And then here's another fun fact that I read about. In New Orleans, there was like cities built on landfills in like minority neighborhoods. It was also found that women living in former landfill sites had a 60% excess rate of breast cancer. That's a really high number. And I know that my mom had breast cancer, and I kind of was thinking about that like as I was doing my research, and like, you know, realizing that like a lot of people who like do live like these sites do have like a lot of health risk. And I kind of just made me think about my home life and like my own family, and that's kind of like why I got into this. This is my project. I decided to like create a mini garden in Detroit. Like it's my church, and yeah, I created a little mini garden, and my focus was pretty much just like educating the public about unfair environmental conditions. So as you'll see like later on the slides, I had the opportunity to speak publicly about my projects at my church, as well as an event at downtown Detroit. And I really decided to like focus on environmental justice because of the simple fact that a lot of people don't know what it is. And then just like looking at my own experiences, it was like, you know, something that was kind of dear to me. And like the recent events about the air quality, and I kind of just started thinking about like pollution and stuff like that, and like how that really impacts like these minority neighborhoods. I decided to create a mini garden. As we all know, like plants hold a great importance to the environment as they naturally purify and clean out our air. Due to like photosynthesis, you know how like the plants absorb carbon dioxide and then release the clean air that we breathe in. So like your plants are very important. And another fun fact I found was that the city of Detroit has the highest levels of air pollution in the state of Michigan, and that Detroit ranks among the top 5% of highest levels of pollution in the country, which is just outrageous. So, and then here's a picture of my nephew. Like as I was teaching about it, like I had like the children like make like little arts projects just so like they'll kind of like understand more like the importance of like plants and stuff like that. And so before I began my project, I did conduct a survey, and I only had like 10 responses, but like that was enough for me. But I asked people in the community if they were aware like these unfair conditions and stuff like that, and if they knew like how important plants were to the environment. And then in these next slides, I kind of go over like my results. As you can see, like in the first one here, not a lot of people knew what environmental injustice was. Like a few people got it incorrect. Like I had like 2 different definitions, and a few people got it incorrect. And then I asked, do you reside near any manufacturing factories? And it was 50-50. Like 5 people said they did, and 5 people said they didn't. So that was pretty interesting. And then, is pollution common where you reside? The orange option represents not sure. So people don't really know if they have like a lot of pollution where they live. And then like some people, as you can see, like the blue, the big shaded area, 80% said they do. When you are driving in the Detroit area, have you seen any manufacturing companies? So this big 80% represents a lot of factories. And then the red, which is 10%, is a few factories, and then orange is no. So I think only had like, it says 10% only said no, and 10% said they've seen a few. And then a lot of people said they've never visited an urban garden. That was 90%. I was really shocked about that, because like I thought that was something that was actually kind of common, but I guess not. And then as I mentioned earlier, like those health risks in the beginning, I asked about those later on, and 40% of people said they've experienced some of that, like eczema, asthma, lead poisoning, or like, you know, any forms of cancer, obesity, bronchitis, and other respiratory diseases and infections. And 60% said no. So that was pretty good. Here's my slide on us like kind of starting the garden, the like buying process. So we purchased the materials. We began telling people about the project and its benefits, and began getting more people involved. So like as I mentioned, I decided to do the garden at my church because it's my dad's church, and I'm able to like maintain it more. And as I'm in college, I know that the community and the church will like help expand the garden, because it is kind of small right now, but they'll help expand it and like, you know, maintain it while I'm away at college. So like that was we had to find the perfect location. And as you can see in this picture, we have bought a few flowers just to make it pretty. And then the planting process, it's here's me and my siblings and my nephew, of course, planting and everything. We tried to have like fun with it and get more people involved. At first, it was a bit of a struggle because, you know, it's hot outside. Like a lot of people don't understand like the purpose of certain things, but we had fun with it. And then here's like pictures of like some of our flowers that we planted. Again, we're still like expanding it as I'm away. We're still like purchasing more things. I'm overseeing it while I'm at college. While we did get a lot done this summer, we still have a long way to go. We are still looking for someone to do a bit more planning to maintain while I'm away in college. We will also be adding more to the garden. And here's a highlight of my summer as well. So like I said, that I did participate. I was under the leadership of Mike Reed under the Zoological Society. So me and my partner Anthony here, we traveled downtown to Belle Isle, where we talked to like hundreds of people about our projects and their impacts in the world. It's called Detroit Metro Youth Day, and it was just a wonderful opportunity to speak out and teach children how to protect themselves and their environment. So I don't know if you can see here, but like I created a board that was kind of like hands-on a little bit. Like kind of I wanted to make it like 3D so like the children will like be more engaged with it. So like I had like a lot of color here. That was just an amazing experience because like just letting like watching the kids kind of understand what we're talking about and like just get more involved with it. It was just really great. I really enjoyed speaking to them and everything. And here's more pictures of us planting and everything. And that really concludes my presentation, but I do want to thank Miss Haley Motley. I'm sorry if I mispronounced that. She has helped me a lot throughout this process. I did have a few struggles at the beginning. Mike Reed, India Hunt, and of course Anthony for just working with me throughout all of this and just doing the best they could and like assisting me and everything. And that pretty much just concludes my presentation.
Well, thank you so much for that. Those pictures of flowers are just beautiful. You've done a great job with the garden. Do we have any questions from inside the room? And I'm keeping an eye on the Zoom chat in case any pop through there.
Thanks so much, Ariel. It's really great to hear about your project. Do you have a sense of how engaged your community is around the garden? Do you think that it will continue to grow and people will continue to plant and expand in it? And are there any plans to let people like plant their own stuff in plots like vegetables?
Yeah, that's actually one thing I did talk to my dad about. I do want more people to be involved with it. So like we're trying to like we're in the works of like gaining more like what's what I'm looking for. We're trying to like make it wider, like more spread out, so like people that know that it's there. So like I'm trying to figure out like how to design it so like you know like wood and stuff like that, and like kind of I don't know how to explain it, like a board like saying that here's our garden and things like that, and like come visit it. And I do want people to feel more engaged with it and like feel free to plant their own things because like it's not just for us, it's more so like for the entire community in that area.
Quick question about the survey. That sounds like a really interesting set of questions and important information to know about the Detroit community. Is that something that the Zoological Society might be able to spread more widely through the community and keep following up?
I feel like it should be. It definitely will. I'll most likely send like that information to Mr. Mike Reed, but I think that it is something that is important to get out to the community and to like build off of. Because like those questions, like I realize a lot of people don't know a lot of information about like pollution or like environmental injustice in general. And I feel like that's very important for us to know, especially living in these communities and neighborhoods.
Thank you.
Everyone, my name is Jace Myrtle. I am a senior at Western Michigan University, majoring in university studies with concentrations in public health administration, environmental sustainabilities, and communications. Also with a minor in general business. It's a mouthful. So this summer, I worked with the city of Ann Arbor on the communication side, working more on understanding about PFAS and their wickedness in our society and environmental systems. My agenda for today is talking about my internship, a little bit more about it, giving you more information about PFAS containing products, an introduction, what are they, their health and environmental impacts, PFAS in Michigan, and PFAS regulation and remediation plans, and your contribution and resources that can help you, and my sources for this project, and also thank you. An internship overview. As I said, I worked with more of the communication side of my internship. So I helped Ann Arbor rewrite their action plan from 2019 that they have not updated yet. So I helped a lot with that and getting new information put in because a lot have been updated since 2019. And I also helped with creating a video for Upstream to help understand more about non-point source pollution on everyday activities like improper care of pet waste and disposing of yard waste and chemicals. PFAS, what are they and what are their possible health risks? Well, PFAS comes in PFAS are an array of chemicals, 14,000 plus chemicals that are from things that are designed to be stain resistant or water resistant, and they are in cleaning products. They are in your clothes. They are in your textiles and carpets. They're in your food wraps, and PFAS have been found to be in your contacts as well. So PFAS are everywhere, and it's something that we have to deal and work with. Why? What are they? Well, PFAS is an acronym for per and polyfluoroalkyl substances. It's a group of over 14,000 synthetic inorganic chemicals which are extensively used and utilized in numerous industries, such as to produce coatings and products that are known and renowned for their resistance to heat, oil, stains, grease, and water. Great things, great products made, by the way, but there are such issues that come with those products that are not so great. PFAS are termed forever chemicals because these are not easily broken down into our bodies. They do not break down in our bodies, and they also do not break down in they're creating persistence. They have a persistence in the environment, and PFAS have been shown to have severe health problems such as cancer, fetal complications, and many more. Environmental impacts. Well, PFAS have been tested in the air, water, soil. It's been tested in humans. It's said that 97% of Americans contain PFAS in their blood, which is a scary amount because that's literally everybody. And it's also been tested in animals. And the PFAS right there on the left side of the screen, that's how PFAS look like in water. It is the foam that is created from all these chemicals being pushed out by certain companies like DuPont. And PFAS have been most studied in these top 10 states: Michigan, California, Alabama, Florida, Minnesota, New Jersey, New York, Pennsylvania, North Carolina, and Massachusetts. And some of the companies that have been responsible for these PFAS contaminations are these companies right here. And actually, there is a current lawsuit with 3M in Michigan right now on PFAS. So it is a very current issue that is affecting us every single day. PFAS exposure and human health. Well, high exposure of PFAS, it creates very horrendous hazards like elevated cholesterol for people who already have cholesterol. That's a major issue. Liver damage, immune system malfunction, and hormonal change increases the risks of kidney and testicular cancer. Studies have shown that a predominant health effects on lab animals to be liver disease, thyroid disease, reproductive disease, and developmental effects, which they study these on farm animals. And so the most common PFAS chemicals out there are PFOA and PFOS, and you can see their chemical makeup right there on the right side of the screen. Michigan and PFAS. Well, this map was developed by the Michigan PFAS Action Response Team and Eagle, and they helped look at the sites where PFAS are most active. There are, as of May 8th and now, there are currently 261 number of sites where PFAS have been tested. And these again are the companies that are responsible for making these products. And these companies are all over your house. They are all over the stores that you shop at. So when you are looking to buy a product, make sure that these companies are not the ones making it. Remediation and regulations. There have been regulations made surrounding PFAS with the Michigan Department of Environment and EGLE as well to establish a contaminant levels for 7 compounds as of August of 2020. And you can see these chemicals in the regulations right there. And the EPA, Environmental Protection Agency, as of 2022, has created a lifetime health advisory levels to measure PFAS contaminations as parts per trillion. To offer protection to people and protections from adverse health effects. And also one thing that I found is that Ann Arbor works very hard to excel past the EPA standards and procedures to protect public health. So the Ann Arbor process for cleaning up the water is 85% of the water comes from the Huron River watershed, and 15% comes from well water, and well water has shown to contain PFAS. And it goes to the Ann Arbor water treatment plant, where it goes through this process of softening with limestone, disinfecting it with ozone, which kills the bacteria, and filters out with carbon. And this carbon filters that they have, it is one of the top technologies that they have to remove PFAS out of the water. And it goes through fluoride additives and additives that they put in the water, and then it gets sent out to their customers. So you might be wondering, what can I do? What can I do to protect myself, as PFAS is everywhere around me and might be in my blood? Well, I would say stay informed, research brands and materials that you use, and test and view your local water quality report. Every year, your city has to release a water quality report. Take a look at that. See what is in your water. What are you drinking? As we are mostly water, it is important to know what's in our water. I believe so. And number 3, suspend use and purchase of products that are stain resistant, oil resistant, just any resistance. Researching what you're using in the products that are going into your home or into your skin as well is very important. Dispose of PFAS products properly. 5, promote awareness and for regulation and remediation. Talking to other people you know about PFAS and bringing it up to your local government as well is a great way to get PFAS on the mind of our decision maker. If you would like to learn more about PFAS and have a visual representation and see how the EPA reacted when DuPont released a bunch of chemicals into the water system of a West Virginia town, you can take a watch at Dark Waters and The Devil We Know. The Devil We Know is a documentary, and The Dark Waters is more like a film adaptation of the movie. It's really great to understanding how corporations and the United States government deal together in environmental issues and also environmental history as well. These are my sources that I used for researching more about PFAS, learning more. And thank you for coming to my presentation. Have a great day.
My name is Anthony Bellinger. I'm with the Detroit Zoological Society. I'm one of the new interns, and this is going to be my project on air pollution community outreach. Next slide, please. There we go. So this is just a little bit about me, but I graduated high school in 2022. I had a dual enrollment program in my high school, so I had early college credits. So with those credits, I took those and went to community college, and I received my first associate's degree within a year in 2023 of spring. Right now, I'm currently an undergraduate student, and I will be attending Michigan State University in the fall. And this is my first internship, so I was very excited to finally have my first internship. Next slide, please. So this is my project description. My project mainly focuses on air pollution and the quality of the air within the Metro Detroit area and air pollution community outreach. This summer, I worked on creating educational learning tools and visual aids, as well as instructional videos for the youth and the community to help them understand these concepts. It also, my project also showcases the demonstration of planting personal gardens, which is one of the popular resolutions to combat air pollution. And some of my goals for this project consist of, again, educating the youth and even some of our adult communities about pollution, and to hopefully convince, inspire, and be effective with my project and make a difference in the environment. And also just make youth excited about learning about pollution and learning how we can impact our environment for the greater good. Next slide, please. So these are some of the educational learning tools and visual aids that I've created. I'm partnered with Mount Olive Full Gospel Church in Detroit, Michigan, and Greater Ecclesia in Romulus, Michigan. I basically create these learning tools and then present it to them during their summer camp events. So as you can see, I created physical coloring books, some digital ones, and then some factory models displaying the major influences and causes of air pollution. And then I also created a YouTube video. As you can see, it's a thumbnail right here, but it's showcasing how to plant plants and the step-by-step processes of it. And then all the way to the right is a visual aid showcasing the air quality index, which is used for reporting daily air quality. And I broke it down and made it less complex for the youth to understand. So I basically, instead of just putting random colors up there, I brung out the little creative side and put plates and put faces to match the types of air quality, and then also putting pictures and index values of our air quality to represent it. Next slide, please. So one of the members of my church, me and him, we started a vegetable garden. He's a part of Mount Olive Church in Detroit, Michigan, and we started a vegetable garden. The vegetables that we have in the garden include tomatoes, kale, squash, cilantro, and potatoes. So those two images up there are of the vegetable garden that we made. And then the garden, the picture at the bottom is the vegetable garden made by the community youth of both churches, which is Greater Ecclesia in Romulus, Michigan, and Mount Olive. And we basically been growing these for quite some time. I would say about April, like the other garden. It was very fun getting all the youth together every week or so often to water these plants and take care of them. And it was a great experience showing them how to take care of plants. And in this case, vegetables, and produce vegetables. Next slide, please. So Detroit Metro Youth Day, this was a great experience because me and Ariel got to showcase our projects. We got to present our projects to a bunch of youth. It was so many young people, so it was enough for every young person to do something. There was food, fun, games, prizes, activities, hands-on activities. It was just a lot of fun that day. So we got to basically showcase some of our visual aids. Ariel got to showcase her board, and I got to showcase my air quality index board. And it was just a great experience teaching some of the youth about our projects and our visual aids. Next slide, please. So the main takeaways, creating these learning tools was a great experience. It was I feel that it's very extremely beneficial for the new generation, especially when many communities, or I've learned that many young people that I was working with really did not have a clear understanding of why certain days they couldn't go outside to play or couldn't be doing outside activities just because of how detrimental the air quality was to them. And then the side effects the health effects it comes with playing outside when you're not supposed to due to the air quality. So I feel that educating the youth on air pollution and our air quality can bring effective awareness, and it has. I've had many parents come up to me and my brother, who's also a member of the Mount Olive Church, and they told us that what we were doing was very least like it wasn't like, I'm sorry, I'm trying to gather my thoughts. They didn't think that many people still did this. Like many people, I guess, would just say the basic stuff, but we tried to break down concepts and make it easier for the youth to learn. That's more what I'm trying to say. And the bottom line is I enjoy working with the youth to make a difference in our environment. These educational tools really benefited the children, and they loved seeing them. They understood them, which was the goal at hand. And I'm just grateful for the opportunity. Thank you. Next slide, or any questions?
Thank you so much. That was great. Questions for Anthony. I was wondering myself, you said that you're heading to Michigan State this fall. What will you be studying, and where do you see that maybe taking you in the future?
So right now, this is my first time ever going into like the environmental field area. I was originally majoring in civil engineering. So now that I did this internship, it's really shifted my perspective about that field. So I may even minor in this field. But right now, I'm majoring in civil engineering. But yeah.
Lots of opportunities in environmental education, as we at Michigan Sea Grant can attest. Many of our coworkers do environmental education on a daily basis, and they're phenomenal at connecting with youth and communities. So can say that's a great thing to incorporate into your education. Other questions from in the room? Hearing none, I think we'll wrap this one up. Thank you very much.